Let's discuss about Archaebacteria, the primitive form of bacteria. So what are Archaebacteria? So Archaebacteria are the primitive or ancestral form of bacteria. So before eubacteria, this Archaebacteria present, which are found in harsh habitat. So these survive in adverse condition in harsh habitat. Why? These bacteria can survive in saline water, in marshy areas, in hot spring areas, in acidic nature areas. Some lives in marshy areas, yes, as I called, not in salty marshes, also in marshy areas. Let's move into the features of Archaebacteria. In Archaebacteria, the cell wall is present like bacteria, but they don't contain no peptidoglycan. No peptidoglycan. They don't have peptidoglycan, that is called as murine. They don't have peptidoglycan, means peptide and glucose. So they don't have peptides and that is polysaccharides. So glycans are polysaccharides. So peptidoglycans are absent in their wall. First difference. Second difference is that they have branched chain lipids present. So normally the lipid chains are single chains, but in the membrane they have branched chain lipids present, and this branched chain lipid helps them to survive in adverse or harsh conditions. Then these Archaebacteria don't have introns. Sorry, they have introns. What are introns? So in normal eukaryotic bacteria, we find exons and introns present. Exons and introns present. So exons are functional genes and introns are non-functional genes. Mostly prokaryotic cells are exonic genes. So all prokaryotic or normal bacteria are exonic genes because they contain exons. They don't have introns. But Archaebacteria do contains introns. What are introns? These are non-functional genes. So they contains non-functional genes in their chromosomes or in the DNA content. So what are the different types of Archaebacteria? One is called as Methanogens, methane producing bacteria. Now Halophiles, which survive in salty area, saline water. High temperature, Acidophiles, high, low pH, acidic condition. So these are three groups of Archaebacteria. Let's start with Methanogens. What are Methanogens? These are special bacteria that produce methane gas from formic acid, carbon dioxide. So these are the bacteria that produces methane gas from specific formic acid or in carbon dioxide. And they live in marshy habitats. They are found in marshy areas, marshy habitats. And they are also, another very important question is, these are anaerobic bacteria. All Methanogens are anaerobic bacteria. And these bacteria are found in the gut of the ruminant stomach. What are ruminant stomach? These are the compound stomach present. These are the compound stomach present. So we find compound stomach, that rumen present, reticulum present, abomasum present, omasum present. So these are compound stomach. So this is mostly present in the cow, buffalo. We find this type of stomach. So in the gut, these bacteria are found to digest the cellulose. It helps in digesting the cellulose of the plant and helps in easy digestion for the animals. And they produce, in return, they produce methane gases. So they produce methane gases, and these methane gases are used as fuel or biogas. These are called as biogas because they liberate energy. They gives fuel. So sometimes it is called as biogas. They use biogas. The best example is Methanococcus and Methanobacterium. The next group is called Halophiles. Halophiles are the group of bacteria that survive in saline water or they are found in salty areas. And these bacteria in the membrane, in the plasma membrane, they contain a purple membrane that contains the chemical that is bacteriorhodopsin. This rhodopsin is mostly present in the retina of rods. In the eye retina of rods, we find this compound is present, but this is now found to be present in the membrane of this particular bacteria. And these membrane normally absorb sunlight and convert the sunlight into ATP. Got it? So these purple membrane, they absorb sunlight and converts the energy into ATP. But these ATP are not used in food productions. Got it? So as they are unable to prepare their own food, so they are heterotrophic in their nature. And the best example is called as Halobacterium and Halococcus. These are the two forms of bacteria. Thermoacidophiles means they can survive at high temperature and low pH means acidic condition. They can survive in acidic condition. High temperature above 80 degrees centigrade they can survive also easily. So these thermoacidophiles found in hot acidic environment. And they survive at high temperature and low pH means they survive at acidic condition. So hot spring, boiling water above 80 degrees centigrade. They can survive above 80 degrees centigrade of boiling water. They also can survive above 100 degrees centigrade also. Normally these bacteria, they oxidize the sulfur to acidic form, sulfuric acid, for which they produce acidic. They're acidic in nature. And the sulfur oxidation do helps in releasing energy. And this energy is used in the preparation of food. This energy is used in the preparation of food. So normally, these thermoacidophiles can prepare their own food. Means they're autotrophic. What kind of autotrophic? Chemoautotropic in nature. Yes, they are chemoautotropic in nature because the energy produced during oxidation of sulfur is used in production of food. So, the chemosynthetic bacteria. Example, thermoplasma and thermoproteus are the best examples. So, this is all about Archaebacteria, or the primitive form of bacteria, or the ancestor form of bacteria. One is Methanogens, another one is Halophiles, and the last one is Thermoacidophiles.